Hello everyone. Our next lecture in clinical pathology is about sickle cell disease. It is the most important and widely prevalent type of hemoglobinopathy, and it is due to the presence of sickle hemoglobin in the red blood cells. The red cells with sickle hemoglobin develop sickling when they are exposed to low oxygen tension, which we will see later. Highest frequency of sickle cell anemia is seen in black race and in Central Africa, where falciparum malaria is endemic. Patients with hemoglobin S are relatively protected against falciparum malaria. There are three different forms of sickle cell anemia: as heterozygous state for hemoglobin S, that is sickle cell trait; as homozygous state of hemoglobin S, that is sickle cell anemia; and as double heterozygous states, for example sickle beta thalassemia or sickle C disease or sickle D disease. Now coming to homozygous state, that is also known as a sickle cell disease or sickle cell anemia. In this condition, there is an abnormal gene which is inherited from each parent. A severe disorder associated with protein clinical manifestation and decreased life expectancy. Now what is pathogenesis of sickle cell anemia? The first and foremost thing is the basic molecular lesion, that is single point mutation, where there is substitution of valine for glutamic acid at 6th position of beta globin chain or beta globin gene, producing hemoglobin alpha to beta 2. So here you can see this is a gene for beta globin chain. At 6th position, glutamic acid is replaced by valine. So that is about basic molecular defect. So the second thing is mechanism of sickling. So there is polymerization of deoxygenated hemoglobin S, which aggregate to form elongated rod-like polymers. So there is presence of sickle hemoglobin within the RBC. So under the condition of deoxygenation, these polymers of hemoglobin S will align and make elongated fiber and distort the red cell into classic sickle shape. So here you can see the pathogenesis in a picture diagram. First is a basic molecular defect where at the 6th position of beta globin gene, glutamic acid is replaced by valine. Then there is presence of hemoglobin S polymers within the RBCs, and under the condition of deoxygenation, these polymers will align and distort the shape of RBC and make the RBCs sickle shaped. So that is mechanism of sickling. But this sickling is reversible initially. So under the condition of oxygenation, there will be a reversal of the sickling. So there will be a normal RBC. But whenever there is repeated episodes of this sickling, the RBC membrane will get damaged, and then there will be a irreversibly damaged cell which will be irreversibly sickled. So this is a 3D picture diagram of a normal RBC, which is round to oval biconcave dish shape, and this is sickled RBC. So third is the reversible irreversible sickling. As I already told you, the oxygen dependent sickling process is usually reversible, but once there is damage to red cell membrane, so there is formation of irreversibly sickled RBC even after they are exposed to normal oxygen tension. Now, which are the factors which determine the rate of sickling? Total hemoglobin concentration, intracellular concentration of sickle hemoglobin, presence of non-sickle hemoglobins, extent of deoxygenation, presence of acidosis and dehydration, and increased concentration of 2,3-BPG in the RBCs. That is about pathogenesis. Now coming to clinical feature. The symptoms of sickle cell anemia begins to appear after 6th month of life, when most of the fetal hemoglobin is replaced by sickle hemoglobin. Infection and folic acid deficiency results in more severe clinical manifestation. So first and foremost presentation is anemia, which is severe hemolytic type of anemia. Second is vaso-occlusive phenomena. Because of obstruction of capillaries by sickled RBCs upon deoxygenation or dehydration, these sickled RBCs will obstruct the capillary blood flow, which lead to microinfarct or macroinfarct. Microinfarcts are usually seen on abdomen, chest, back and joints, and they will lead to pain crisis. Macroinfarcts are seen in spleen, which leads to autosplenectomy, in bone marrow, which leads to pain, in bones, which leads to aseptic necrosis or osteomyelitis, in lungs, in kidneys, in brain, or in eye or skin, and these results into anatomic and functional damage of these organs. So this is a picture diagram. Here you can see that sickle cells blocking the blood flow in compared to normal RBCs. Here sickle cells are blocking the capillaries because of their shape. This is a picture diagram. Here you can see in the A figure there is a normal spleen, and above that there is a infarcted spleen which is reduced in size, and microscopically you can see that there is, in figure B, there is red areas which are congested area, and then there is pale areas which are fibrosed area. In figure C you can see the sickle shaped RBCs in the sinusoids of spleen. So this is about spleen. So repeated infarction in the spleen lead to autosplenectomy, a fibrosed spleen, a non-functioning spleen, that is known as autosplenectomy, which is seen in sickle cell anemia. Then constitutional symptoms. In addition to anemia and infarction or vaso-occlusive crisis, impaired growth and development and increased susceptibility to infection because of markedly impaired splenic function is not uncommon. So these are the features of sickle cell anemia. You can see the features of anemias, severe anemia like fatigue, breathlessness, then jaundice as a sign of hemolytic anemia. Then there are signs of vaso-occlusive crisis and pain crisis like back pain, leg and arm pain, joint pain, etc. Now coming to laboratory findings of sickle cell anemia. You will see moderate to severe anemia. The hemoglobin is reduced. The blood film shows sickle cells and target cells and features of splenic atrophy such as presence of Howell-Jolly bodies. Usually these Howell-Jolly bodies, the RBCs which contain Howell-Jolly bodies are phagocytosed within the reticuloendothelial system or spleen. But because here there is autosplenectomy, the features of splenic atrophy such as presence of Howell-Jolly bodies within the RBC is seen on peripheral smear. Then you will have a positive sickling test. Now what is sickling test? In sickling test, a sample of venous blood or capillary blood is collected. Then the blood is mixed with the reducing agent, for example sodium metabisulfite. That reducing agent will initiate or induce sickling in the susceptible cells, and the results can be viewed under a microscope after 20 minutes. So we have to leave the mixture of blood and reducing agent for 20 minutes, and then we have to make a smear from that and we have to examine it under the microscope. The test is simple, quick and used to identify the presence of sickle hemoglobin. So here you can see in a normal patient the test is negative, the RBCs are of normal round to oval in shape, while in case of sickle cell anemia the test is positive. You can see the sickle shaped RBCs in the microscope. The other test is known as sickle solubility test, in which there is again a mixture of blood and reducing solution is made in the test tube, and the sickle cell anemia patient will show a turbid appearance of that mixture, so you cannot see the lines through the test tube, while in normal patient there will be a clear solution, so you can see the lines in the background through the test tube. So that is known as sickle solubility test. In this test you don't require microscope, but the sickling test is more sensitive than the sickling solubility test. So this is a picture diagram. How can you see the sickle shaped RBC in the peripheral smear? Again here you can see the sickle shaped RBC in the center. Again sickle cell RBC as well as normoblast in the peripheral blood smear. Then you can go for electrophoresis also. The hemoglobin electrophoresis will show no normal hemoglobin A, but show predominance of hemoglobin S and up to 20% of fetal hemoglobin. So this is a band of electrophoresis. Here you can see the last is a normal phenotype where there is adult hemoglobin, hemoglobin A type. You can see the band of that. Then in the middle there is a sickle cell anemia phenotype. You can see the band in the region of sickle hemoglobin, and the uppermost is a sickle cell trait or heterozygous form. Here you can see the band in the region of adult hemoglobin as well as in the region of sickle hemoglobin. So that is all about homozygous state of sickle cell disease. Now heterozygous state, that is also known as sickle cell trait. Only one abnormal gene is inherited in compared to homozygous state where the two abnormal genes are inherited from each parent. No significant clinical problems are seen in these patients except when they become severely hypoxic, they develop sickle cell crisis. Laboratory findings, there is no anemia and they have normal appearance of RBCs, but in state of hypoxia, sickle cell crisis develop, and the diagnosis is made by demonstration of sickling done under condition of reduced oxygen tension by an oxygen consuming reagent, that is sodium metabisulfite, or you can go for electrophoresis, which reveal 35 to 40% of total hemoglobin is sickle hemoglobin. Then last is double heterozygous state. These conditions involve the combination of sickle hemoglobin with other hemoglobinopathies. Most common among these are sickle beta thalassemia. All these disorders behave like mild form of sickle cell disease. Their diagnosis is made by hemoglobin electrophoresis and separating the different hemoglobins. That is all about the sickle cell anemia. Thank you very much.